today is noradrenaline is a stress-associated metaplastic signal of GABA synapses. This was published in Nature Neuroscience this May by a Canadian group. First, we need to overview how stress response goes down to the body. Adaptation under stress condition is a priority for survival to all the animals. And stress can be defined as actual or anticipated disruption of homeostasis or anticipated threat to the well-being of the animal. And stressor-related information from all major sensory systems like interoceptive cues like blood volume or osmolality, and exceptroreceptive cues like odor from predator or preparing for journal club like that is delivered to the brain, and brain recruits neural circuits or neuroendocrine system to minimize the net cost to the animal. And the physiological response to stress aims to maintain physiological integrity even in the most demanding of circumstances. And stress response recruits a efficient and highly conserved interlocking systems. This is very natural because stress means threat to the life to the animal. So the response should be go down very smoothly and very fast. So the interlocking system is well established in the animal system. And these stress response recruits autonomic nervous system and HPA axis system. Autonomic nervous system provides the most immediate response to the stress exposure. For example, sympathomedullary arm can rapidly insert cause high rate increase or something like that. But in the HPA axis system, hypothalamic pituitary adrenocortical axis, it results in elevation in circulating glucocorticoids. So here you can see the schematic picture: hypothalamus and the pituitary gland and the adrenal glands. So this two-step hormonal mechanism is very slow relative to ANS system synaptic mechanisms. And in this hypothalamus, there exists paraventricular nucleus, shortly PVN, and PVN is a principal integrator of stress signals from all over the body. And this PVN houses distinct population of neurons that project to the median eminence to autonomic targets in the brainstem and spinal cord. And it is heavily innervated by GABAergic inputs. And in the PVN, there are many PNCs, parvocellular neuroendocrine cells. And there are two types of PNCs, type 1, and the types of the PNCs can be discriminated by electrophysiological fingerprints like low threshold spikes and robust T-type crushing current and such like that. So, and on the other hand, beta-adrenergic receptors is recruited in the stress response system. And epinephrine, noradrenaline, epinephrine is bind to the beta-adrenergic receptor, and a subsequent cell signaling occurs. And PKA have important function in this system. So in this paper, the authors try to confirm that a single stress can alter the GABA synapse property onto PNCs to undergo activity-dependent synaptic plasticity. Because in previous studies, exposure to a single stressor can change the neuroendocrine responses to future challenge of the stress. So this might mean that stress can be learned and prepare for the further future challenge of the stress. So the authors try to figure out the response in the GABA synapses in the PVN. So they gave rats immobilization stress. This is rat restrainer. Rat is very packed in this restrainer for 30 minutes. It must be very stressful. After 30 minutes, the rat was sacrificed, and the hypothalamus slice was obtained, and high-frequency stimulus was given after immobilization. 4 high-frequency stimulus at 100 Hertz for 1 second with 20-second interval. With naive rats, HF induced no potentiation of evoked iPSC, but in the stress rats, immobilization stress can cause potentiation of eIPSC after HFS. And then the authors try to give different protocol of stimulus, like for half a second stimulation. The initial stimulation was for 1 second, half a second stimulation, and longer stimulation for 4 seconds. And you can see here, half a second stimulation was compared to the naive one. The potentiation occurred, but it was not significant. But and the 4 second stimulation was significantly enhanced. And they gave another stressors other than immobilization stress. In the slice, they gave electrical stimulus that is 10 Hertz. Initially, it was 100 Hertz. 10 Hertz and for 10 seconds, which mimics better mimics the natural stress inputs to the PVN. And predator odor was also used. I guess it was fox urine was used as a predator odor. And forced swim test was also used. In these 2 cases, eIPSC potentiation was occurred significantly, but in the forced swim, eIPSC potentiation occurred, but not so significantly. So far we have known that the immobilization stress and other kinds of stress can cause a potentiation of eIPSC after high-frequency stimulation. So, on the other hand, beta adrenergic receptor activation during stress is necessary for unmasking subsequent LTP-GABA was asked by the authors. So PRO and ISO was introduced to the slides. PRO is propranolol, and it is a beta AR antagonist, and ISO is isoproterenol, and it is beta AR agonist. So before immobilization stress, PRO, beta adrenergic receptor antagonist, was introduced, and after that, HF was given. Then you can see here, the potentiation was blocked here. But in the naive rats, when you activate beta AR and then give HFS, potentiation occurs here. But here, transient, very large potentiation occurs here, but it just subsidizes very shortly after that. And after that, the authors try to find the contribution of noradrenergic inputs to GABA synapse priming. They used teach-cree mice in this task. So they expressed channelrhodopsin or EYFP or channelrhodopsin or EYFP in the PVN region, and when they gave light, blue light, 5 milliseconds for 10 hertz, I guess, and when the light is on, the EIPSC potentiation was occurred, but when there's no light, EYFP case, there was no potentiation occurred. And when beta adrenergic receptor was blocked by PRO, also the LTP GABA was blocked. And previous studies have shown that PKA activity downstream of beta adrenergic receptor is important for intracellular signaling recruited in the stress response. So the authors try to check the PKA activity in the study. So they gave naive rats the 8-bromos EAMP, which is PKA activator, before high frequency stimulus. And you can see here, if you activate PKA, the potentiation occurs even in the naive rats. And when you use beta receptor agonist ISO and PKE inhibitor TK 57220 together, the potentiation of EIPSC was blocked. And so far we have see that immobilizing stress can cause LTP GABA after HFS, and beta adrenergic receptor activation is recruited for LTP GABA, and PKA downstream of beta AR activation is also recruited in this stress response. And then there are other studies saying that group 1 mGluRs are included in this stress response. So the authors try to specify which mGluR is responsible for the changes in the stress response. So they used multiple mGluR antagonists and agonists to see the effect. So here MCPG group 1 and 2 mGluR antagonist was used, and if you block group 1, 2 mGluR, potentiation didn't occur. And CPCCO FAET, J&G, like this. These mGluR1 subtype specific antagonists, these affect different region to the mGluR1 mGluR1, and these could block the potentiation of EIPSC, but MTAP, mGluR5 specific antagonist, didn't block the potentiation of EIPSC, and this GABA AR antagonist was also used, and it couldn't block the LTP GABA. So group 1, 2 mGluR and group 1 antagonist, and mGluR5 specific antagonist can show that only mGluR1 is recruited in the stress response. So we now know that mGluR1 is recruited in the stress response. So the authors try to activate mGluR1 by DHPG, and they show that it is sufficient to induce LTP GABA. So this is apart from HFS induced stress, just activating mGluR1 to see if just activating mGluR1 can cause potentiation in the PVN cells. So DHP was given by BET application, and sometime after that, you can see stress rats show potentiation in EIPSC, whereas live rats shows no potentiation. So, and then the others try to ask if DHPG induced potentiation, and former HFS-induced potentiation share the same downstream mechanism. So they first applied DHPG and wait until the potentiation occurred, and then they gave HFS here. They did this because if two modes of stimulation share the same downstream mechanism, then HPG also already induced a potentiation of EIPSC, so they may have recruited all the entities below the downstream the signaling pathway. So then there would be no entities to activated by HFS. So you can see here after the HPG enhances the EIPSC, then even after HFS there was no potentiation in EIPSC. So we might say that this DHPG induced the EIPSC increase and HFS induced GABA share the same downstream mechanism. So then they had to connect the beta AR activation and mGluR1 activation together. So they wanted to know whether beta AR activation causes the functional upregulation of mGluR1 after immobilization stress. So they demonstrated by blocking beta AR by ISO PRO. When you gave PRO beta AR antagonist before immobilization stress, then after that you can after you activated mGluR1 by the HPG, there's no potentiation occurred. So inactivating blocking beta AR before immobilization stress could block LTP GABA. So and one thing to note here is that if you apply DHPG before ISO, meaning activate mGluR1 before beta adrenergic receptor, then this kind of effect doesn't occur. ISO is beta adrenergic agonist. So if mGluR1 is active, then after that, even you block the beta AR, then it has no effect on that. So the temporal sequence of this event should be beta AR activation, then mGluR1 activation. So the authors try to see the synaptic properties of this GABA synaptic effect, and they looked at pair-to-pairs ratio and square coefficient of variance here. The top panel shows the PPR and square coefficient of variance here. Before HFS, the baseline PPR and square coefficient was similar, about the same between naive rats and stress rats. But after potentiation during LTP GABA, coefficient of variance increased and PPR was remained just the same. This panel is for stress rat slice. So PPR means alteration in PPR means changes in release probability of neurotransmitter, and coefficient of variance could mean that changes in coefficient of variance could mean that changes in release probability or alteration in number of functional synapses, or it could be both. So here, PPR was just the same, and square coefficient variance was increased, so it could be mean that number of functional synapses is increased here. And this linear regression plot shows that amplitude of EIPSC and coefficient variance have strong positive relation. And after that, they try to see the postsynaptic effect of this stress response. And it is known that intracellular calcium level and KMK2 activities is important for this stress effect. So they use BAPTA, which is a calcium chelator. So they applied BAPTA and used the EIPSC, and they could see that the potentiation was blocked by BAPTA. And AIP is a KMK2 inhibitor, and also KMK2 inhibitor could block the LTP GABA. So intracellular calcium level and KMK2 also have their own function in this stress response. Here, BONTC, botulinum toxin C, is a toxin responsible for which responsible for snare-dependent exocytosis. So if you apply BONTC, snare-dependent exocytosis is blocked. So when you apply the toxin, the EIPSC could drop here. So they did this because in previous result, if the number of functional synapses is the cause of this increase in LTP GABA, then because number of synapses increase, the spontaneous response of iPSCs should increase too, because there are many number of synapses. So they also checked spontaneous iPSC frequency and amplitude. And amplitude was just the same, even though all the calcium-related treatments on exocytosis block. But as iPSC frequency was enhanced after stress response, and also blocked by BAPTA, AIP, and BONTC.
I have a question. So what's the functional meaning of blocking snare-dependent exocytosis? What does it do?
Releasing a neurotransmitter.
So basically you are blocking neurotransmitter releasing.
Yes. So after that, they use this method of substituting calcium to strontium. I didn't really got the purpose of these methods, but before and after HFS, calcium in ACSF was substituted by strontium, and in the stress and the purpose of this using strontium is to see. Okay, so calcium causes synchronous fast onset response, but on the other hand, strontium causes slow onset asynchronous response, we say. So in here you can see when you apply strontium before HFS and after HFS, you can plot here. Inter-event interval can be treated as frequency of the response. Compared to before HFS, after HFS, the frequency of the response increased, and this is blocked by Bo and TC. As you can see here, the graphs just match the same. Yeah, and they use Bo and TC and DHPG together. So if Angular 1 is functionally upregulated, they use this GABA AR is the Bo and TC to see if the GABA AR insertion is increased. So DHPG would make more receptor to be to respond to the DHPG. So they applied DHPG, and stress reps show the potentiation, but Bo and TC block the effect of DHPG. So this is the end of the data. And so in this paper, the authors show that beta adrenergic receptor can be used as extracting stress information that PVN receive from all over the body. And then beta adrenergic receptor response goes down to pKa level, and pKa activity is necessary for LTP-GABA. And in the synapse snare-dependent exocytosis, it recruited in the synaptic mechanism of this LTP-GABA, and overall beta AR-dependent metaplasticity at GABA synapses is recruited in the stress response. Is the meaning of the paper. Yes.
Could you explain about the physiological meaning of this phenomenon?
Physiological meaning.
What is function of this plasticity in behaviorally or?
PVN is a center of receiving stress response from all over the body. A sensory stimulus came into the center of this receiving sensory response from all over the body. And beta adrenergic receptor, in previous studies, there were no paper looking at the beta adrenergic receptor response and this PVN receptor together. So beta adrenergic receptor response can extract information of the stress, because beta adrenergic response is also a recruitment stress response. So PVN receive all the data, and beta adrenergic receptor can extract which information to use, because beta adrenergic receptor signal and PVN response come together, then they can discriminate which stress response to.
So PNC is inverted to amygdala, and in previous in another group, the circuit from PNC to amygdala is important for this fear expression. So I think that there can be answer for you.
This group published 2 papers at the same time doing almost the same thing. The next one is showing LTD GABA. So
In nature neuroscience,
yes.
they the same group
Yes.
from.
But different receptors and subtly different.
Right.
LTD is also excited in GABA or.
Yes. They say that also that LTD is stress. So that's kind of metaplasticity also, but they are, in that paper, they're concerning glucocorticoid receptors.
LTD is by stress, immobilized stress.
So yeah, so stress is kind of causing metaplasticity to induce LTD.
In 2011, they published the same, almost same paper with this, but in that paper, this LTP is not covered. LTP is stated for.
I don't know, but in PBMism mostly is covered inputs goes in, so they could see any effect.
I haven't seen the supplementary information from this paper. Did they check any glucocorticoid receptor?
No, there was only 4 figures in the supplementary. There was no.
So they're explaining two different mechanisms inducing LTP and LTD,
Yeah.
but stress is both inducing and causing metaplastic change. And in LTD, that paper, I think it was mGluR 5 dependent. This is mGluR 1 dependent.
Yes. The stress response is primarily for homeostasis, so
Why this group do that? Don't do behavior experiment.
Is it well established that PKA can regulate mGluR1 causing metaplasticity?
I don't know if it's well established, but there are papers.
Actually, to me, it's not familiar, even though I searched a lot about PKA. PKA regulated mGluR1.
But there are reference here, so I guess. But in the discussion, they said the functional role of PKA is to be elucidated further. So
And if the PKA is really essential factor in this paper, then is it possible other mechanisms could involved in rather than the beta-adrenergic receptors?
if PKA is important, it could be, but I don't know. We can know that.
But they just only made the conclusion that noradrenaline is
Yes,
really important
Yes.
in the toxicity.
yes.
They check effect of NMDAR receptor.
In another paper of Nature Neuroscience by the same group, they did that.
They examine in postpartum mechanism. They check 10 times to alpha calcium, the effect of calcium. However, in case of PKAs, they just best applicate the inhibitor or activator, right? So pre and post synapses are affected by the PKA inhibitor or activator. Does that mean the PKA is both? Well, I wonder if they tried some just examine the activity of PKA in post-sensitive, whether it is only effect to post-sensitive or the PKA activity is also essential to use presentive modulation. I wonder what do you think about? So then in PK, in presenting role, PK is needed to both present and post-sensitive.
Yes, but I don't know. They argue that only number of functional synapses as number of functional synapses. So exactly how presynaptic and postsynaptic effect can cause these LTP GABAs is not explicitly said. So but it may have function if we disrupt PKA activity in presynaptic region.
My question was kind of crude, so I ask again. Because they test the effect of PK with other molecule in personality, but they show there is some effect, but they didn't check whether the PK specifically applies to personality. So I think, is there any possibility the PK only applies to personality? If they examine, it will be.
Yeah, but yeah.
Well, in this paper, they exclude possibility that
Yeah.
presynaptic region is involved in the metaplasticity.
Because PKA they mentioned here is under downstream of beta adrenergic receptor, PKA. So
Are there some methods that can measure stress level?
if you give stress, firstly there are immediate response. If you threat the animal, it can be measured by freezing level, but also hormone, glucocorticoid, which is stress hormone, can upregulate in the plasma. So doing stress experiments, people take blood out from the animal and measure the level of glucocorticoid hormones. That is commonly